Start. So this is the humble nematode worm, scientific name Connectome elegans, or C. elegans for short. It's a common model organism in both genetics and neuroscience due to its small size. It's about the width of a hair and up to 1 millimeter in length, and it has under 1,000 cells in its entire body, 989, 302 of which are neurons. It has the smallest count of neurons of any known organism. So it's a pretty interesting place to look if you want to start understanding how nature constructs brains. Now, back in the 80s, there was this massive project undertaken in the UK where dozens of graduate students painstakingly sliced up a worm and traced on these electron micrographs each neuron. You can see some of the neurons are labeled in this image, and produced this connectome, which shows all 302 of the neurons and how they are connected. It's pretty obtuse. There's not really a whole lot you can actually make out of this because it doesn't tell you what the connections do. If you wanted to start writing a program to animate this image with each neuron's level of activity, you would have trouble, as I did 3 years ago, when it came time to write the method that would actually update the values based on the values from the previous point in time, because you don't even know whether the connections that are marked in the connectome are inhibitory or excitatory, let alone how strong they are relative to each other or what kind of characteristic timescale they have. In order to learn this information, we not only have to know where the connections are anatomically, but what they do functionally. And that means instead of studying a dead, sliced up worm, we need to study a live worm. And we now have the tools to do that using fluorescence, because the worm happens to be optically transparent, and we can put in optogenetic molecules which bind to calcium, which is a proxy of neural activity, and fluoresce more when there's more calcium. So they're brighter when more things are happening there. And if we take the hypothesis that each of these neurons, fairly simple, not many dendrites or anything, is actually only representing one variable at a time, which is an extremely common assumption in the land of artificial neural networks, then we should be able to extract all of the information we want if we can control and image each of the neurons in this optical method. Now, in order to do that imaging, because the neurons happen to be arranged in 3 dimensions, we have to use some clever optical techniques. This is called light field microscopy. Typically, when light reaches the sensor plane, which would be here, and one marked D, the angle of the rays is information that's lost. Whatever angle they come in, they activate the sensor, and you get the same result. But if you put a micro lens array at the sensor plane and put the actual sensor behind that, the result is you get these little micro images where the position of intensities in that micro image tell you what direction it came from, and that allows you to actually invert the optical process if you have some bounds on the distance of light sources from the focal plane, and you get a 3-dimensional thing. So, in order to actually explore and visualize this data, because it's quite large and existing software is basically non-existent, we built a Meteor App with WebGL. So this is what the actual raw data looks like. Each of these little circles represents one of the micro lenses. And now we can switch to light field rendering view. We can actually move this around. And these are videos. We're capturing several frames at a time. So you can see the worm kind of crawling along here. Each of these frames is like 4 megabytes, so it takes some time to load. But each of them is fully 3-dimensional. Now we're working on algorithms to extract automatically what's going on in each neuron. Any questions? Well, this is a rendering of a single frame, and this slider here is where we are in the video. These sliders are just for gain and gamma, which helps somewhat with the contrast. But essentially, we're taking they're 2-dimensional frames, but representing 3-dimensional data at each time point. And
through intensity of the image.
Yes. I mean, they're grayscale images, 16-bit. Yep.
302 is such a reasonably small number. How close are we to, I mean, like, could you actually write this out as code? Do we understand all the connections between all the neurons? sorry if this is so
we do not understand them. That's why I'm doing this.
So do we know the connections between them, and do we know the individual function?
So we do know which pairs of neurons have a synapse, and there's one bit of data we have, which is whether it's a chemical synapse or an electrical synapse. Electrical synapses are symmetric, the chemical synapses are one way. So you could write out, and there are files which you can freely download from various sources, all of those pairs, do graph viz on them. But you can't do a dynamic simulation because there's no dynamic information.
In the electrical synapse, do we know anything about the size of the electric potential? Nanowatt, smaller than nanowatt, microwatt?
The potential is in millivolts. Yes, but we don't know how they're relative to each other. It's just a general biological fact that potentials are in tens of millivolts.
How many different neurotransmitters are there?
8. Hopefully that information about the neurotransmitters we don't need to extract explicitly, they'll sort of come out of the, using different neurotransmitters will result in different coefficients, and we'll measure those coefficients directly.
So what's your deal, dude? Are you like, is this like your hobby, or is this like, you get a t-shirt? So this, you get a grant, or?
Yes, I have a grant. I am an independent scientist.
Cool, thanks so much.